All right, probably my most requested topic, protein synthesis. And remember that protein synthesis is divided into two parts. It's probably one of the most difficult concepts that the IGC expects you to understand. So just be patient, okay? Work it over in your mind. There are some good videos out there as well. God help me as I try to draw this. So there's two main parts, all right? The first one is transcription. And just knowing these are key vocab terms, all right? So just knowing them will help you. So the first one is transcription, and the second one is translation. Now, transcription is, so the whole idea of protein synthesis is that you have your DNA in the nucleus of a cell holds the genetic code for your proteins, right, to produce the proteins. But the problem is that the ribosomes, which is where the proteins are made, are in the cytoplasm. So if you want to get your code from the nucleus to the ribosomes, then you need to make a temporary copy of the genetic code. And that's what the transcription is. The translation is the actual production of proteins, but the transcription is, the whole idea is that you're basically making a copy of the genetic code, a temporary copy, if you will. And you do that, all right, what's happening is that the DNA is copied and you end up with the production of mRNA, messenger, and the M stands for messenger. It's literally being used as a messenger, right? It's going into the ribosome and telling and carrying the message for the DNA. So so DNA to mRNA, and that's messenger RNA. All right, this all happens in the nucleus. The entire process of transcription happens in the nucleus. So that's one of the easy ways to tell them apart. Transcription is in the nucleus, C and C, if you want to remember it that way, right? Transcription, nucleus. Okay, I feel like I'm reaching, but that's the idea. All right, translation is basically taking that copy of the genetic code and actually making proteins. So this is DNA. Producing that polypeptide chain. So this is where you're going from mRNA to the actual protein or polypeptide chain. And this happens in the cytoplasm. And the other part you need are ribosomes. Okay, that's an on. Doesn't look like it, but it's on ribosomes. All right, so ribosomes, another keyword that you need to make sure you know. Now, how does that happen? Okay, so let me get you a nice picture drawn, and we'll talk about transcription first. So this is transcription. And so this is your DNA here, your double-stranded DNA. And what I've shown is that where the transcription actually takes place, the DNA parts and separates, okay? And so what I haven't shown is over here, you would have the complementary bases, right? So you would have C, A, T, et cetera, going down this side, but I'm not going to try to crowd up my drawing, okay? So the strand that is being actually used to transcribe from is called the template strand. Sometimes it's called the antisense strand, but you can just call it the template strand. All right, the DNA. And so what happens is is you have mRNA. So this is the nucleotides, right, on the DNA. So the first thing that happens is the DNA unwinds, or sometimes it's called unzipping, and that exposes the bases. All right, so that's what I've shown here, is that the bases are exposed and they are ready to form complementary bases. Now you do need to know, I'll do a short video on comparing RNA and DNA and what a nucleotide consists of, but for right now, these are your nitrogenous bases. There are 4 options, right, G, T, A, and C, and you need to know which one pairs with which. And so what happens is that now that the bases are exposed, you have mRNA nucleotides that come in and will match up. So G goes with C, so you have a little nucleotide here. Now T goes with A, okay? And so mRNA doesn't have T, it has a U instead, uracil, so that joins up. This would be with a G here. I'm losing my backbone. Another G, another U, et cetera, et cetera. Hopefully you guys get the idea. All right, and so this is the mRNA strand that's being formed. All right, these are mRNA nucleotides. Each individual one with a single letter is an mRNA nucleotide, and then that begins to form a strand of mRNA. So you have RNA nucleotides enter. And basically they use what we call the complementary base pairing rule to pair up. So they pair up with complementary, make sure you spell it right, complementary with an E, not saying nice things, but instead matching, okay? Pair up with complementary bases and form an mRNA strand. All right, and so that's what's forming here. And so this keeps on forming until it reaches the end of the gene, which could be, 20 bases, it could be 200 bases, depending on the gene. And then the mRNA strand detaches and leaves the nucleus. Leaves the nucleus. Okay, so then this mRNA will separate from the DNA, and then the DNA will close back up. All right, so that's transcription. There obviously is more that's going on, but the level of detail at the iGCSE, this is plenty. All right, so then I'm going to do translation next. So for translation, so this happens in the cytoplasm. And the first thing that always happens is that the mRNA binds to a ribosome. All right. And the ribosome starting at a start codon. All right. So this is the other thing. So the mRNA, we talk about the mRNA in units of 3. So this is a triplet. It's called a triplet codon. And so 3 bases on the mRNA, the triplet codon, that's going to equal 1 amino acid. There are about 20 amino acids that you can code for, and there are about 64. There are 64 possibilities that you can have in terms of a sequence of a triplet codon. And so the idea, because you can have any of the 4 bases at any of these 4 spots, so 4 times 4 times 4. That's where you get 64 from. And the idea for the triplet codon is that the order of these the bases within the triplet codon will determine the which tRNA binds, right? And so I want to attempt to draw a tRNA over here. Looks like a kind of like a weird 3-leaf clover, all right? And so the idea is that it has a space to carry an amino acid on it. Right, everybody see that? So this is my tRNA. And this is an amino acid. Remember that proteins are made up of amino acids, right? So this is like a single building block of a protein. And it has an amino acid on one side, and then on the other side of it, it has what is called an anticodon, which is a sequence of 3 nucleotides again. And the idea is that, so let's make this match. Let's see, it would be GUA, all right? And so this anticodon, the sequence here on this anticodon determines which amino acid the tRNA is carrying. And so the idea is that after the mRNA binds to the ribosome, the tRNAs are going to enter and they're going to match up. The codon and the anticodon are going to match. So here, CAUGUA, those are complementary bases, right? And so this tRNA comes in and will sit right here. So the tRNA enters the ribosome, and it's carrying a specific amino acid that is dictated by its anticodon. And of course, the one that binds is then dictated by the codon on the mRNA. So it's carrying a specific amino acid. All right, and so that one's going to enter, and then a second one will enter. And so the anticodon on that one would be, hopefully you can see, would be CCA. Yes, for the GGU. So this is the second triplet codon here. All right, and so the second one is going to then enter. And when the second tRNA enters, then what happens is, just draw this real quickly. You have this, sorry for the terrible drawing, and then you have another one here, And so you have these amino acids, right? These are my tRNAs. I know they look like some sort of angel that's been run over by a bulldozer, but still. All right, so these are your amino acids. All right, and so the second tRNA, when it enters, then a bond forms between the two adjacent amino acids. The ones that are next to each other, that's what adjacent means. All right, so a bond is going to be formed right here. All right, and then you're going to have a third one come in, the same thing. You'll have a bond formed over here, and the first one leaves without the amino acid. So the amino acid kind of falls off the tRNA once it's bound to the next amino acid, and so then you're going to get a growing chain of a polypeptide chain, a growing chain of amino acids, which is your protein. So then, let me see where I'm still writing. Yes, okay. So the first tRNA leaves. The ribosome kind of moves down by three bases. A third one enters, right? A third tRNA enters. Another bond is formed, and the process continues. Another bond between the amino acids forms, and the process continues until the ribosome hits a stop codon. And a stop codon is a three-letter sequence that doesn't have an amino acid corresponding to it, and so the ribosome falls off, and the protein and the long polypeptide chain is complete. Continues until a stop codon is reached. There we go. Okay. So that is translation. I'll do another video if you want to, if you're wondering more about the details of the structure of mRNA and DNA, I'll do another video on that.